now for the third line of defense. The third line of defense is highly specific, and it is also much more sophisticated than the first line and second lines of defense. Here we're dealing with B lymphocytes and T lymphocytes. We have two types of third line of defense: antibody-mediated immunity, which is also known as humoral immunity. The antibody-mediated immunity is largely driven by B lymphocytes, and the purpose of this is to make antibodies that will target the pathogen. The next type of third line of defense is cell-mediated immunity. This is largely driven by T lymphocytes. The purpose of cell-mediated immunity is to kill cancer cells and virus-infected cells. Both of these types of immunity are highly specific, and they must actually be primed by an antigen-presenting cell before they will target a pathogen. They are not part of the general surveillance or general immunity. Before we go into the process of antibody-mediated immunity, let's take a look at the 5 major classes of antibodies. We have IgA, IgD, IgE, IgG, and IgM. The word Ig stands for immunoglobulin, which is another word for an antibody. Antibodies are proteins. IgA antibodies are found in bodily secretions such as saliva, tears, mucus, and breast milk. IgD are a special kind of antibodies that are always found attached to B lymphocytes. IgE antibodies are involved in allergy reactions. IgG is the most abundant type of immunoglobulin or antibody, and this is very important for fighting off infection when you are exposed to a pathogen a second or third time. These are very small, and so the IgG can actually cross the placenta. The anti-Rh antibodies belong to this group. IgM antibodies are very large. These are the largest class of antibodies, and so they cannot cross the placenta. The blood types A and B, the antibodies against them, anti-A and anti-B antibodies, cannot cross the placenta, and they belong to the same group. Let's imagine that a virus has entered the bloodstream. This is the antigen on top of the virus, and the goal is for the B cells to make antibodies that will exactly match this antigen. However, we have millions of different kinds of B cells, each with its own antibody. So to find the correct match between the antibody and the antigen can take many days. So let's imagine that we pick the red antibody as the chosen B cell and the antibody that correctly matches the antigen. This stage of antibody-mediated immunity is called antigen recognition, and it can take several days. By then, the virus is already multiplying and infecting the body. Even though the correct B cell and its antibody has been chosen, the antibodies cannot be made until permission is received from the helper T cell. So a different kind of B cell presents the antigen to the helper T cell. It kills the pathogen first and then presents it to the helper T cell, which produces a chemical called cytokine. Cytokines have the ability to stimulate the production of antibodies in the next few steps. This stage is called antigen presentation, and the cell is called an APC, or an antigen presenting cell. And the purpose is to get permission from the helper T cell. Now that the B cell has permission, the chosen B cell then multiplies. It forms a clone of itself. Once it forms a clone of itself, it will be ready to differentiate and make antibodies. So these are the chosen B cells, the ones that have the ability to produce the correct antibody matched to the antigen. And this stage is called clonal selection. Once many new B cells are made, the chosen B cells are made and they multiply, they now differentiate into two types of cells. One kind of cell is called the plasma cell. These are the pink cells that you see here, and they have the ability to actually make the antibody. What you see in blue is the rough ER, and if you remember, the rough ER make proteins that leave the cell. Antibodies are proteins that leave the cell, which is why you see the rough ER here. The other type of cell that is the memory B cell, and these cells have the antibodies attached to them so that they will be able to recognize the antigen again when there is a future infection. So these are memory B cells that will circulate in the body. And this stage is called differentiation, and this is stage 4. The last stage in antibody mediated immunity is the production of antibodies. As you see, this is a plasma B cell and with its rough ER, very functional, and it's producing antibodies. The Y-shaped red structures that you see here are the antibodies, and they have the ability to target the antigen so that they may be marked for destruction by other cells, by other white blood cells. Antibodies do not directly destroy the pathogen. They do it indirectly by targeting the pathogen in a variety of ways. So let's look at them one at a time. The first is neutralization. The antibodies surround the pathogen, they find the antigens on the pathogen and attach themselves to the pathogen. So here's the pathogen shown in gray. It has antigens on it, and the yellow antibodies that we see here attach themselves to the pathogen, thereby rendering them ineffective. The second way is agglutination. It has the word glue in it, which means it makes the pathogens stick to each other. The antibodies attach themselves in such a way that the pathogens stick to each other, thereby rendering them ineffective. The third way is precipitation. It's like sweeping up the antigens that may also be toxins in the blood, sweeping them up and making them more visible. So here are the antibodies attaching themselves to free antigens and making them more visible. The fourth way is to activate the complement proteins. That's one of the most important functions of antibodies, is to turn on the complement proteins. The complement proteins, once activated, can make holes in the pathogen and cause death or destruction. The neutralization, agglutination, and precipitation make the pathogen and the free antigens or toxins produced by the pathogen more visible to phagocytes. In this way, phagocytosis is enhanced. Complement proteins, once activated, can make holes in the pathogen, causing the pathogen to be destroyed. We call that cytolysis. Remember, we said that cytos means cell, lysis means destroy or break down.